So we have been all discussing so important things. No, we are all here involved in thinking about humans' health, animal health, and we have not been discussing, but this is also very important to consider. This is an important argument about the 17 objectives of the sustainable development from the United Nations. So control hunger is one important objective, and we have to accomplish these objectives until 2030. So 8 years from now, right? So everything we are discussing, I think, is also part of this. We have to think about this. To produce food for the world, right? We are 8 billion people, and the more we think about this, to produce good food and sustainable food for the world, the better it will be. So I'll just present something very quickly. This was a presentation I did for Israel government. Well, some things you have already seen. So I will pass this, right? The total GMO approvals in Brazil by CT and Bio were 104 genetically modified plants, including corn, cotton, 104 genetically modified plants, until April, 50 recombinant vaccines for thank you, for dogs, cats, poultry, pigs, equines, and tilapias, 2 genetically modified mosquitoes, 1 moth, 1 genetically modified Spodoptera frugiperda, 1 Atlantic salmon from Aqua Bounty, 42 genetically modified microorganisms, mostly for production of ethanol. All this information is available at CTN Bio's website. Is publicly available. You can have all this information if you like. These are the approvals, right? Mostly plants, but also microorganisms, insects, etc. Total of 201 approvals until April. We have some more. And these are the 2 transgenic mosquitoes that have been approved by commercial release by CTN Bio. These are the genetically modified gene therapies approved by CTN Bio. All of these, they are being used now. In 2021, these vaccines for COVID-19 have been also analyzed and approved by CTN Bio, including Pfizer, Janssen, Sputnik, AstraZeneca, and the one from Biomanguinhos, Fiocruz, Brazil. Well, but we had a boom, right, about genetic engineering in the years 72, 73. We see here Princess Diana. It's an old issue of time. And we have these 2 authors, and Paul Berg was one of the most important that got the Nobel Prize. And then in 2013, we have this that also changed the world. So we are all thinking about this. We are almost 10 years now, and we are all, it challenged all of us, right, with this new technique, and mostly for the regulatory activities. This has been very, very challenging for all of us. Right. And well, so we started to think at CTN Bio, and our legal definition of a GMO is based on the recombinant DNA, RNA, and molecules, right? And this implies the linking DNA sequence to a plasmid or to a vector, and to cut DNA molecule in a cell and inserting this construction in the cell so that it can express in a living cell. So this is the DNA, the recombinant DNA technique in which our law was based. But this is the transgenesis. We have the donor, we have the receiving organism, and we have the new combination of genetic material. This slide has been given to me by our friend, Agostina. She authorized it by my use of this slide. And so we have a new construction here, and this is a GMO, right? This is a GMO, and it must be regulated as such. Now we have the genome editing, which is the modern biotechnology application. We have the organism as a receiver, and you apply a genome editing technique like CRISPR, TALENs, etc. And then you modify here, you very specifically, you cause a mutation in the cell, and there's no combination of genetic material. There's no material that remain in the cell, right? This is the desired situation, right? No combination of genetic material, just cut the DNA and then you repair a section of the DNA of a gene, for example, and this can be modified or repaired by the own cell enzymes. So, so this is a gene-edited product without new genes, without plasmids, without pieces or sequences from a vector. This is what we expect to be a gene-edited product. And then based on this, we created the normative resolution number 16. There's a paper that counts all the story here by Alexandre Nepomuceno and other people. If you like to know, this is the normative resolution. It includes what are the types, the product, how the product must be. We agree, no, that this normative resolution is mostly dedicated to plants and microorganisms, but it also can include viral vectors, right? Live beings with recombinant viruses expressing genetic modifications and implications without host genome modifications, and other RNA-dependent DNA methylation. So there are many techniques here which are accepted as a new precision breeding innovation techniques. These are examples. And then, in order to submit to apply for registration or approval of CTN Bio for a genetically modified organism, you have to send a letter of inquiry, and it must contain some information, which is identification of the genetic technology, taxonomy classification, risk classification of the parental, the genes that are manipulated, genes or manipulated genetic elements, and their specific functions where applicable. This is all in Annex 2, and the company must fill this form to send with the letter to CTN Bio. Then genetic strategies used to produce the modifications, molecular characterization of the result in the recipient organism, providing information related to number of copies, manipulated, location of the manipulated region in the genome when possible, and identification of the presence of off-target genetic modifications, if any. So this, the company must feel to submit the letter. The product of expression of the genomic regions manipulated, described in detail when applicable, and in relation to the product, so the process and the product, this is the product evidence of the absence of recombinant DNA, RNA molecules through the use of molecular methods. If the product containing DNA, RNA for topical systemic use has recombinant ability to insert into the target species and or non-target species, whether the product referred in the submission has been commercially approved in other countries. This is important too. If the product uses the principle of gene drive, that may enable the phenotypic change conferred to the potentially disseminated throughout the recipient organism's population, and then explain the care to monitor the organism, so what to do, and how the possibility of target effects of the technology that may be present in the product was evaluated. So important, gene drive and off-target effects. So in the analysis, so we have to send a letter with description of the parental product, the technique employed, and precaution regarding gene drive and off-target effects in the principle of the case-by-case analysis. And the conclusion is whether a product is a GMO or not, according to the Brazilian biosafety law. So according to our system, to our law, SDN 1 and 2 are considered non-GMO, and SDN 3 is considered a GMO, right? So this is what we have. And then, as I told you before, CTN Bio approves or not. If it is approved, it goes to the specific agency or ministry for regulation, for registration, right? This is the plant, the same thing. And so until April, where I prepared this presentation, we had 10 yeasts for the production of ethanol, waxy corn, sugarcane, 2 varieties from Embrapa, low raffinose soybeans, canine parvovirus vaccine, 1 fish, the aqua bounty tilapia, the sperm from an Elori cattle, edited myostatin gene, exogen, and the hornless cattle agro partners consulting. This has been canceled. And then this is the tilapia. Tilapia is an organism that comes from Africa, the Nile tilapia. And it had been modified, its DNA has been modified through CRISPR-Cas9 and 26 base pair deletion in the gene of interest, which is the myostatin 1 gene. This myostatin 1 gene is, myostatin is a growth factor that arrests the growth of the muscle. So if you delete part of it, it doesn't arrest the growth of the muscle, and the result is a bigger tilapia. So the edited tilapia shows an increase in muscle mass and greater weight, and the yield of the fillet in comparison with its counterpart without editing. So the process has many confidential parts, so I cannot speak too much about it, but this is the construction, and it has been considered as a non-GMO by CTN Bio, and it had been considered also as a non-GMO by Conabia, right, in Argentina. So this is what we have analyzed all the process. It has been analyzed by one colleague from the University of São Paulo, and then the plenary session of CTN Bio approved as a non-GMO. There's a story about the hornless cattle, if you don't mind. I'm gonna briefly comment on this. It is Bury from Allison, the University of California. Bury was developed by combining by teams, no? And it had, this is the control, and this is the hornless. And it had, it's a story that has been published in our FAPESP journal. The FAPESP is the research foundation. So it was, the construction was the introgression of the Celtic allele. So it is the replacement of 10 base pair deletion, replaced by 212 base pairs construction. And okay, they submitted the process for approval of this bull's semen to CTN Bio. It has been analyzed very carefully, and it has been approved as non-GMO. However, when this process was analyzed by FDA, they found plasmid, a piece of short sequence of plasmid among this animal's cells. So it has been considered like this GMO animal. So this is the story. This is the laboratory. Here we talked a lot with Heather about this. And it is so sad because, you know, I'm going to stop here. It is so sad because what are the implications for the safety of these animals if you have a short sequence of plasmid? This is something very sad. However, it is the law. And the Axeligen, in fact, withdraw this request for CTN Bio, so the process is stopped there.